Welcome to all. This is a video lesson on transcription in eukaryotes. So before going into the actual process of eukaryotic transcription, it would be helpful to recall transcription in prokaryotes and the basics of what happens in prokaryotic transcription. Will help understand this process better. So to begin with, transcription is a process of synthesizing functional mRNAs or RNA molecules using DNA as template. And this template DNA from which RNA molecules are derived is what we call a gene. Okay, so when this RNA happens to be an mRNA, then it proceeds to the next stage in the gene expression, which we call translation, where mRNA gets read or decoded into polypeptides or proteins. So in general, when we talk about transcription, the process is same in both eukaryotes as well as prokaryotes. Both transcription processes involve the same stages, the same steps, namely initiation, elongation, and termination. And both processes involve the same enzyme. By saying the same enzyme, I don't mean the enzymes are same in their structure or complexity, but they are both the same type of enzymes. They are both RNA polymerases. RNA polymerase is an enzyme which is capable of making RNA molecules using DNA as template. We have seen another version of the same type of polymerase in DNA replication, where we call it DNA polymerase. DNA polymerase is an enzyme which makes DNA molecules using DNA as template, whereas RNA polymerase makes RNA molecules, or rather it adds RNA molecules using DNA as template. So both processes involve RNA polymerase, both in prokaryotes as well as in eukaryotes. Transcription involves RNA polymerase as the chief enzyme, the main enzyme or major enzyme. And in both cases, there are recognition sequences which help this enzyme and other proteins involved in the process to recognize that there is a recognition sequence is present in both cases, and in both cases there are several protein factors which are involved in assisting this RNA polymerase during transcription of RNA from DNA. So these are the things which are common to the process of transcription which happens in prokaryotes as well as in eukaryotes. So coming to the, this is the image showing the actual stages of transcription. So like this, you have a recognition sequence, which in order to recall what happens in prokaryotes, we call these regions as Pribnow box. We have two regions, one in -10 and -35. One is called Pribnow box, this region, which is closer to the start site. Then you have another sequence in -35 region. And these regions are recognized by something called the sigma factor, which is a subunit of the RNA polymerase enzyme, which is found in bacteria and other prokaryotes. So these sequences are only called recognition sequences, and they are often found to be conserved sequences or consensus sequences. They are the same for they are almost similar for the same organism across different genes and across different organisms also belonging to the same kingdom. So which is why it is called a consensus sequence, and these are the stages. First, there is a recognition happening, recognition of the promoter elements. Okay, these recognition sequences constitute what is called the promoter region of a gene. Okay, so these promoter sequences are recognized by RNA polymerase, or rather the sigma factor of the RNA polymerase, and thereafter transcription, other transcription factors join this process, and transcription is initiated. And after that, the RNA polymerase begins to move along the template strand. Okay, they begin to move along the DNA, and when this initiation complex moves along the DNA molecule, the region of the DNA which is being read opens up, and it appears like a bubble, and which is what we call as transcription bubble. Usually it is less than 20 nucleotides long. Only that much of the DNA is opened up at any given time. So, and the mRNA is being synthesized by the RNA polymerase, which you can see as a yellow strand here. And then termination happens. And when you take bacterial transcription, termination happens in two ways. One is called row dependent and the other is called row independent transcription. And usually there is something called an inverted repeat sequence, which is located in the fag end of the DNA molecule, fag end of the gene, which is being transcribed, and that region forms a hairpin loop-like structure on the primary transcript of the mRNA, and that is what triggers the RNA polymerase to stop transcribe. Okay. So this is the overall process of transcription which happens in bacteria, which I mean in a very simple recollection, for the purpose of recollection in a nutshell. And this concept of sense and antisense strand is also the same for prokaryotes and eukaryotes. The strand which is actually containing the codes is called coding strand. The coding strand is also called the sense strand, this is what runs from 5' direction to 3' direction on the DNA. Now we are talking about the DNA. And if you take this strand, this is the sense strand, and this is what contains the 3-letter codon sequence. Already, as the viewers can understand, the mRNA is made up of 3-letter codons, and each 3-letter codon corresponds to a amino acid, and this is what we call genetic code, the details of which will be dealt with in detail in another video of translation process. So here, when you take this sequence, the mRNA, the codons and the mRNA come from, the source of the codons come from DNA. So in the DNA, mRNA is single-stranded, so there is no problem. But when you take DNA, there are 2 strands. So which of the strands actually contains the codons? Which of the strands has the information, Dina? Then the 5' 3' direction strand is what is actually containing the information as it is in the mRNA strand. It is just a copy of what is there in the mRNA. So we call it sense strand, okay, or the coding strand, because it contains the codons. Okay. So when we want another copy of the codons, it is only sensible that the strand which is opposite to the coding strand needs to be transcribed, because the strand which is opposite to the coding strand is in 3' 5' direction, and that strand has a complementary sequence set on it. The sequence on the 3' 5' strand is complementary to the sequence on the 5' 3' strand. So when we transcribe the 3' 5' strand, we get a 5' 3' direction mRNA, and that mRNA has the sequence in the order of what is already there in the coding strand. Okay, so this concept of sense and antisense strand also applies to both prokaryotes and eukaryotes. There are no changes. The strand which is meaningful is actually the strand which is in 5 prime 3 prime direction that contains the codes, but the strand which is actually used as template for performing transcription is the strand opposite to the coding strand. So it is called antisense strand, or rather it is also called template strand, because that is only used as the template for making RNA molecules. So these concepts are same. So fine, these are same. So what are the differences between transcription processes which happen in prokaryotes and eukaryotes? What are the variations? The first variation is that the length of the DNA which is getting transcribed is enormously large when compared to that of a bacteria. In bacteria or other prokaryotes in general, the genome size itself is very small compared to that of any eukaryote. Even the simplest eukaryote has a much longer DNA, much bigger genome than a bacterium. So since the amount of DNA, the length of the DNA that needs to be transcribed is longer than that of any prokaryote. So therefore, the rate of transcription needs to be much faster. There is a higher rate of transcription in eukaryotes than in prokaryotes, because for example, if you take a prokaryote, you can have a gene within a span of 2 to 3 kilobase pairs, and therefore the rate of transcription can be hundreds of base pairs per second. That much is sufficient, and within minutes it is possible for a bacterium to transcribe completely transcribe a gene and to get a complete primary mRNA transcript. It takes few minutes for a bacterium. Whereas if you take a eukaryotic organism, the length of the DNA is so large, it runs to kilobases, thousands of kilobases, and therefore the rate of transcription also needs to be equally high. And in spite of such a high rate of transcription, it takes anywhere between several hours to even days for a eukaryotic cell to make a mRNA molecule. So that's how big the difference lies in the size of the genome between a eukaryote and a prokaryote. And then other important differences include transcription factors. The number of transcription factors, or when we say factors, we are referring to proteins which are assisting the process of transcription. So there are n number of transcription factors and elongation factors in eukaryotes, which is very large when compared to the number of proteins involved in prokaryotic transcription. And there are RNA polymerases, are also many RNA polymerases are present in eukaryotes. At least 3 different types of RNA polymerases are there, whereas in prokaryotes there is only one type of RNA polymerase which is capable of transcribing all the genes present in a prokaryotic organism. And then another reason why this there is a variation, this is a very important fact which contributes to the variation, is that there are introns. Intron sequences are present in the eukaryotic DNA, but the concept of intron is completely absent in a prokaryote. There are no introns in prokaryotes, whereas eukaryotes contain non-coding regions called introns lying in between the coding regions of a gene. So this is what we call introns and exons. Introns are non-coding regions, whereas exons are the coding regions of a gene. So even within the span of a single gene, it is possible that there are several exons separated by several introns lying at certain intervals. So these introns also contribute to the length of the DNA molecule that needs to be transcribed, and also later propose the problem of their removal. Introns need to be removed from the mRNA molecule later. So because they are not needed, they are non-coding sequences, and therefore they need to be removed before the mRNA molecule goes into protein synthesis or translation. So this is another major difference. And then there are DNA binding proteins called histones, which is a very familiar term to most biology students. Histones are proteins which are responsible for winding and binding of the DNA. They are DNA compacting proteins that are responsible for compacting the DNA while it is not in use. When we say not in use, we are referring to a DNA which is not being transcribed or replicated. So when DNA is in such a resting state, it is tightly wound and it is found in the form of nucleosomes, or what we call broadly as heterochromatin. So this heterochromatic DNA is unpackaged and loosened up for transcription and replication. So but however, histone proteins are not completely removed from the DNA for the purpose of transcription. So histones are present, but they unwind as and when transcription happens. So dealing with histones requires special enzymes and proteins during transcription. So this is another big variation from prokaryotic transcription. And here another important thing is eukaryotes have nucleus. Prokaryotes do not have an organelle system. There is no separate nucleus. So everything, transcription, translation, everything happens in one go in the same place in a prokaryote. Whereas in a eukaryote, it's not like that, because there is a compartmentalized system. You have a cell, you have compartmentalized organelles. So this transcription happens in the DNA molecule, and therefore this process is taking place inside the nucleus. So this mRNA has to come out of the nucleus after transcription is over. So that this variation is a very prominent one between prokaryotes and eukaryotes, because since prokaryotes do not have this compartmentalized organelle system, prokaryotes can simultaneously perform transcription and translation. It's like while transcription is going on in one direction, the mRNA transcript that is coming out of the transcription bubble on the 5' end is attached to ribosomes, and translation is initiated even before the process of transcription is completed. So it is possible only in a bacterium, only in a prokaryote, because there is no separate nucleus. There is no endomembrane system to separate this DNA molecule from any other part of the cell. But that is not the case in a eukaryote, because DNA is well protected inside a nucleus, and there is a nuclear membrane, and special arrangements have to be made to bring the mRNA out of the nucleus in order to proceed to translation, because translation happens in the cytoplasm or on the endoplasmic reticulum, but not inside the nucleus. So we have to bring the mRNA out of the nucleus, and only then translation can be started. So performing simultaneous translation and transcription is an impossibility in eukaryotes, but it is a possibility in prokaryotes. So these are the changes which we observe in general in the process of transcription. So which, to put it in a nutshell, the complexity is what matters. Prokaryotic transcription is simple, but the same stages are happening in eukaryotes in a much more complex manner, on a larger scale. So in addition to this, so these 3 points are very important. These 3 processes are unique to prokaryotic transcription. So unique to eukaryotic transcription. One is 7-methylguanosine capping, the other is poly A tailing, and the other is intron splicing. So these three processes are completely absent in a prokaryotic system. The mRNA that is transcribed in a eukaryote undergoes a special process of protecting its 5' end. Okay, and that is called 7-methylguanosine capping. So we'll see in detail about that later. So that is one phenomenon, and the other is poly-A tail, which happens on the 3' end of the mRNA transcript. The 3' end is polyadenylated before it is allowed to go out of the nucleus. The primary mRNA transcript, which is synthesized from the DNA, is capped at the 5' end and it is polyadenylated in the 3' end. After these two processes are over, the intron sequences which have been transcribed need to be removed from the primary mRNA transcript. Not only intron removal is important, introns are removed and the exons are reunited. That is what is very important. Which is why this process is called splicing. It's not just about cutting, it's about cutting and rejoining of the exon sequences on the mRNA transcript. So these three processes are unique to a eukaryotic transcription system. And these three processes are usually called post-transcriptional modification, but in reality, 7-methylguanosine capping and polyadenylation happens along the process of transcription. As and when transcription is going on, the mRNA transcript undergoes these modifications, but splicing takes place after the process of transcription is over. It is a separate phenomenon which goes on in the nucleus. So this is an image to give a rough idea of comparison between prokaryotic and eukaryotic transcription. As you can see here, transcription is going on, and before transcription is terminated, here the 5' end of the mRNA, nascent mRNA, the new mRNA which is made, is being translated with the help of ribosomes simultaneously. And because there is no nuclear membrane or any membrane for that matter. But when you take a eukaryotic system, it is not like that. You have a separate nuclear membrane, nucleus, and therefore the transcription process is going on here, and after that is terminated, this 5' capping, polyadenylation is happening, and after that these unnecessary sequences, non-coding regions which are present in the DNA cannot be avoided during the transcription. So they are transcribed, but they are an unnecessary burden when it comes to translation. So they need to be removed, and that process is happening, and that's called splicing. So intron removal is happening, and after that these mRNAs need to be safely exported out of the nucleus and into the cytoplasm, where ribosomes are waiting for these mRNA molecules so that it can perform translation. So these are the fundamental differences which lie between transcription process of a bacterium and a eukaryote. So we will go into the details of initiation, elongation, and termination process of transcription in a eukaryotic system in the upcoming videos. Thank you.